before we get started? Anything at all?
Is there a spot where it's explicitly written what we're supposed to have read or completed before we come to class each day?
So is there a spot where it's explicitly stated what you're supposed to do before coming to class each day? It's usually going to be the last slide of the previous class's set of slides. In general, my expectation is that if I've assigned a chapter of the manual to read, that includes everything that's in that chapter, both the written content and also the videos that are linked in that chapter, and attempting the exercises that are in that chapter. Think of them as homework assignments. Except, unlike homework assignments, they're not to be turned in for points. It's practice opportunity so that when we come to class, like we're doing for the first time today, if you attempted some of those exercises and you have questions about them, then we can discuss them in class, where everybody gets to benefit from that discussion. We can work through the answer together if you want, and that's how this class is meant to operate. Is that sufficiently answered?
Yes.
Awesome. Yes?
When I was working on the Google Sheets, I had trouble finding the C-Briggs percentages for the AT, C, and G.
Yes, and that was deceptively designed to do so. So I'm glad that you were willing to say that in class, because that's usually one of the obstacles to getting this structure of class to work, is for people to say, yeah, I had a problem doing that. But we will definitely go over that in class, and if I don't answer your concern about that, please let me know. Perfect. Yeah?
For the HTLV part,
Yes.
I couldn't find a T percentage, but instead a U percentage. Is it like...
That's another excellent question. So when we get there, if I don't answer that, remind me. But yes, percent. So for the aficionados in the room, if you're there already, the percent thiamine is essentially the same as percent uracil for the HTLV sequence on the Google Sheet. So if you don't know what I just said, that's fine. We'll cover it again when we get there. I just wanted to make sure I answered your question before we got there. Anything else? Any concerns, questions? Yeah.
The last two questions in the Google Sheets, there was some kind of discussion question, and then there was instructions on how to turn it in, but I didn't see any of that.
I apologize. Thank you. So the last part of that assignment on the Google Sheet was something about turning it in, which I actually didn't intend you to, so don't worry about that part. But thank you for pointing that out.
When I was doing the reading, some of the links didn't work.
Yes. Oh, and boy, you guys are sharp, guys and gals, and everybody else too. I'm bad at using, colloquial language, so slap me if I start doing something that's sexist or anyway. One of the videos that was linked in the introduction to the syllabus, or the introduction to the manual chapter, the how to use the manual video, I think, didn't work. That was my fault. I accidentally broke the URL when I was editing that. So I actually I fixed it, and I've reposted it as of like 45 minutes ago. So if you do want to go back and watch a video on how I organized the course manual, you can. It's not essential for having done for class today. It's just in case you want to know how I laid it out and how I intend students to use the manual. But totally up to you. But thank you for pointing that out. Anything else? Okay, well, please raise your hand during class if you have a question. I'm always happy to answer questions during class. After class today, I do have a little bit of time afterwards if anybody wants me to hang around and talk about anything in particular. So to get us all in the science mindset, which is part of what we're doing in this class, it's not just about genetics, it's not just about content, it's about thinking like a scientist, and especially, you don't need my permission to be creative in how you analyze and interpret data, but scientists sometimes don't think that it can be a creative process, but it can be very creative, because even coming up with hypotheses, you have to be creative about how to disprove somebody's idea. So a couple years ago, I found this picture somewhere on the interwebs, and I thought, I don't know if that's true. So quick discussion. Does anybody have any comments? Does this distribution of lengths of colored pencils prove that this guy or gal's 3-year-old daughter that her favorite color is or is not pink? That's a hypothesis. Let's see if is it supported or can we refute it? Sir?
I think you could make evidence for both ways. Like it could be her favorite because she's saving it for last, or light blue could be her favorite because she's using it the most.
Okay. So light blue So the longest pencil might be the favorite color. Maybe she uses it less because she wants to be able to use it more in the future. Or the opposite interpretation. Maybe blue has been used the most just because she colors a lot of pictures with sky. Right? Doesn't prove that blue is her favorite color, it just proves that she colors a lot with blue. So as we go through the semester, the whole point of this brief discussion is anytime you see something, don't necessarily take it at face value. Be willing to critique it. Be willing to say, I don't believe that until you really prove it to me. That's my request. If you're willing. So a few of you did notice that I had already opened up this exercise at the start of class, but if you haven't yet, please log back onto Socrative, do a quick introductory quiz. Again, the room name is Ross Genetics at Socrative.com or your Socrative student app. And as usual, what I'm doing today is I'm asking you questions. This is just scored for completion. 1 point, if you finish it and you've put your name in, then I record you as at first being present today and then also having completed this activity. But the utility for me is that I get to find out if there was something about nucleotide composition and nucleotide structure, the content you should have read for today, that people generally don't still understand, in which case we'll spend more time going through that in class today. While that's happening, if anybody has any other questions or concerns, I'm happy to talk through those. If you're still working on it, that's fine. You've got time to finish up. I'm just going to switch over to looking at a few of the results. So let's see. Cytosine is a purine. Well, it looks like we're leaning towards false. What's cytosine? Pyrimidine. And you remember that is because, or the easiest way to remember this, cytosine has a Y, and so does pyrimidine, and so does the other pyrimidine, which is thymine. Yep. So that's false. Cytosine is a pyrimidine. Okay, that's close enough to good to not have to go through in detail. Nucleotides can be categorized by how many hydrogen bonds they form with other nucleotides, either 2 or 3. We'll talk about that a little bit more in detail in class today. Yes, adenine and thymine nucleotides do form 2 hydrogen bonds between them. Again, we'll talk about that a little bit in class, not at length. And, so if one strand of DNA has 10 nucleotides, then the opposite strand will have how many T's? So you're right, absolutely. How did you figure this out? Quick summary. Somebody shout it out. If A's bind with base pair of T's, then every A on this strand will be opposite A T. So there's one A, there'd be 1 T, 2 T's, 3 T's on the opposite strand. Again, I'll touch on that briefly. Just wanted to check. So that tells me that for the most part, we're not going to do a ton of review of nucleotide structure today. A little bit, not too much. So thank you for that. Okay. Little mindset. So I'm big on sort of trying to address deficit mindsets. Like I've tried this before and I'm not good at it, I'm not a science person, those sorts of things. Which, if you did read this paper, I hope you got something out of it. You'll know this was one of the additional readings in the textbook manual or the course manual. And yes, it was about doctoral programs, it wasn't about undergraduate programs, but the points are still valid, I think. And that is that, yeah, science is hard, but if you find it easy, then you might not actually be applying yourself as much as you should be. Right? So just because you feel stupid, you shouldn't feel stupid. That means you're pushing yourself. And that's the point I'm trying to get everybody in the class to, is actually meeting and exceeding your own standards for what you think is successful. But of course, I'm here to help you through those processes when you run into obstacles. So please, 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 if you have the opportunity and you run into an obstacle, ask me. Come see me in office hours, make a separate appointment. That's my job. That's what I see myself here doing for you. And the beautiful thing about science is, regardless of what happens, sometimes there isn't a right answer to every question. If you haven't written that down, you probably should. In this class, and in science in general, there is not always one right answer. Think exams. There's not always one right answer to every question. And that's the process of science. We test a hypothesis. Sometimes we get a clear answer. Sometimes we get an answer that supports no particular perspective. Sometimes we have to try harder, come up with new experiments, and try again. So as we go through class, you'll probably encounter some situations where the answer isn't really clear. That's usually where students, and I sometimes, feel a little bit uneasy. But that's okay, because that's the way science actually works. So the way that the class is going to go today is we're going to spend about half of the remaining time, maybe 15 or 20 minutes, working a little bit more on DNA structure, base pairing, hydrogen bonding. It's going to go pretty quickly. And again, these are the vocabulary and the tasks for today's class. If you look at these tasks, do those look familiar? Did you see those somewhere else recently? I think they were on one of the surveys that I had you take. Every one of these chapters of the class manual has tasks at the very beginning. These are my clues to you about what's going to be on tests. If it's something that is important enough for us to spend time doing, these are things that I'm going to try to assess. Can you do that on a test? So that's why I wanted to give you that survey that was due at the start of class, one of them, due at the start of class today. That was a preview to all of the things that we should, I hopefully will be able to bring you all along with and help everybody be able to do by the end of the semester. Where you self-identified, I think I can do this, I'm not sure, I don't understand if I could do this, I'm sure I could do all of this perfectly, that survey. Right. That was a starting point, and now you've seen now you've seen all the tasks basically for the class, the goals, the objectives, what we're going to do over the course of the semester. And so those are the three topics we're going to focus a little bit of discussion on today. The last 10 or so minutes, I want to give you a little bit of anonymous feedback from the two surveys that were due. Does anybody have any questions about DNA structure labeling? Any of these? So this was one of the exercises. Can you label all of these topics? So this is vocabulary. I don't usually spend a lot of time on it unless absolutely there are questions.
Difference between a base pair and nucleotide?
Right. So the base pair, let's start with, quickly, hydrogen bonding. So here we have a couple of nucleotides. There, I'm circling in black. Okay. One of them's a purine, one of them's a pyrimidine. Base pairs are always one purine, one pyrimidine. Where do the hydrogen bonds between these nucleotides form? And how many of them are there? Let's start with the easier one. This is why organic chemistry is a co-requisite or a prerequisite for genetics. So if I zoom out, which direction do the backbones of the two strands of DNA run? This is a molecule of DNA. There are two strands. So I'm just asking, one of the questions is backbone. What's the backbone of DNA? That's where the phosphate groups are. Those P's with all the O's around them, phosphates with oxygens surrounding them. Those are the backbones of the two strands of DNA. So we have one strand here and the other strand here. Okay, so these are two covalently bound chemical molecules. These are all covalent bonds in the backbones. Right. Those are weak bonds or strong bonds? Covalent bonds. They're stronger than, what's the type of bond I'm asking about up here? Hydrogen bonds, which are weaker. They're more easily broken. Okay, so those two backbones of DNA hold together under most circumstances. The weaker bonds can be broken. What's an easy way to break hydrogen bonds? Heat. So normally at room temperature, say, or body temperature, DNA is pretty stable. There are hydrogen bonds that form between, I hope this works, hydrogens and nitrogens and hydrogens and oxygens between the nucleotides. So now that is the base pair. It's a pair of nitrogen-containing bases that form what otherwise would be called maybe a step. If this looks like a ladder, there's the two rungs of the ladder, and then each base pair creates sort of a step on the ladder. And those two backbones are held together by those, in this case, 3 hydrogen bonds between those two nucleotides. That's a base pair. So we could fill in all the rest of the hydrogen bonds. And if we heat up this molecule of DNA, that adds energy to the system. And when we add energy to this system, that can cause the backbones to shake with the increased energy that's available in the system. And then those hydrogen bonds can break apart. Those bonds then can be severed. And instead of a double helix, where all of the base pairs are connected by these hydrogen bonds, you have two no helices. They don't form helical structure when the two backbones are separated. You have two single-stranded pieces of DNA. So that's the backbone, bases, base pairs, nucleotides, strands, location of hydrogen bonds. Wow, that was fast. Any other follow-up questions? One thing that I really hope you like in the course manual, tell me if you don't, is that part where I say this is what you should not focus on content-wise. And one of those things was structures of organic molecules, I think. Right? I'm not going to ask you to draw uracil or adenine or thymine or guanine or cytosine. That's organic chemistry. That's okay. So the last thing, and probably the most important thing, which no one has asked about and we haven't talked about yet, is, for genetics, the polarity of these molecules, which is something that in my experience, including my own personal experience when I was in your seat, was my least favorite part about studying the structure of DNA. Where the 5' and the 3' go? The polarity of DNA. So what's the easiest way to know where a 5' end of a molecule is? So there's something about hydroxyl groups. Okay, so if I erase some of this so I can actually see the blanks that I had. Right, there's a phosphate at one end of each molecule of DNA. There's a phosphate here at one end. There's a hydroxyl, an OH group at the other end. So this is something you just have to memorize. The phosphate group's the 5' end. And the hydroxyl group is the 3' end. So you may have learned that in high school, you may not have. You may have learned that in biology 1 or not. But that's just a rule you need to memorize. So on the other strand, on the other backbone, what happens when you label the phosphate 5? Where is that? Top or bottom? Bottom. And this is why there's an OH there at the top. This is why DNA is sometimes referred to as, the A word. The two strands are anti-parallel because one goes 5' to 3' top down, the other strand goes 5' to 3' bottom up. It's always a characteristic of a DNA double helix. One strand has one polarity, one strand has the other polarity. I'm going to go off on an unplanned adventure, just briefly, but I'm seeing that glazed look in some of your eyes. Like either I've had this before or I have no idea why the hell this is important. What does this mean to me? So why is polarity important to geneticists? Why do we have to know the polarity of a molecule?
I think when you're listing the nucleotide sequences, you have to first put 3' or 5' to know which way you're reading it.
It all is about which way you're reading it. So, to make that a little bit clearer, maybe, I hope, say we've got this sequence of DNA, ATAGC. That might be part of a gene that encodes a protein, and so it's important to know which direction the cellular machinery will grab onto the DNA and read the sequence of nucleotides, right? DNA, all it is information. You've probably heard these analogies before. DNA is like a book. A chromosome is like a book. DNA is like the letters in a book. If you open a book and you read it backwards from end to start, not just by word but by letter, it's not going to make any sense at all. Let's see. If I can try to read, let's see, what's Socrative backward? Evitarkos, right? That means nothing to us. I'm reading the words backwards by letter. So if we're going to understand what the DNA encodes, what messages it has for the cellular machinery to read, what the protein sequences are going to be, we have to know, is this actually ATAGC or is it CGATA? Because those two things mean totally different things, both to us when we read English or any other language, and to the cell when enzymes read the DNA. So the convention is we always write 5' on the left side, 3' on the right. Convention. So if I ever write a sequence of DNA and I don't explicitly state which end is which, just assume that we're reading, as the cell would, from left to right, 5' to 3'. So that is why polarity is so important, and it will be on tests. Any questions? So think about this for, let's say, a minute. We've got 4 different strands of DNA. Each of them, as I just said, start with 5' on the left. That doesn't really matter for this exercise. Which one of them will denature or melt at the lowest temperature? Take a minute. Talk to a neighbor if you want. I'd like to get a little bit of noise. So if you want to talk to somebody, convince them that your answer is correct, and we'll come back together in 1 minute, talk about the answer. Go ahead. Any questions? Questions? Okay, I've heard some good conversations. So what were the things that you were looking for when you tried to answer this question?
The
the amount of the different nucleotides, like A and T and.
Okay, so the number of different types of nucleotides. We also call that base pair or nucleotide composition. How many A's, how many T's, how many G's, how many C's are in each of these molecules? By the way, geneticists often represent DNA by writing one strand of sequence like I did up here. Why do you think that is? Why not write both strands? Because what's missing up here is I didn't write in, for example, the opposite strand of each of these. So you have to remember that these are double helices. With my horrible tablet handwriting, I'll try to fill this one in. So T's pair with A's, G's pair with C's. But I only gave you the top strand of each of those double helices. Why? It's an easy answer. I mean, obvious answer.
There's only one possible pair that can go.
Right. So there's only one sequence because there's only one way the top strand can base pair with the bottom strand. So basically, it's an efficiency thing. Why should I bother writing the bottom strand when you can figure it out on your own by writing a T underneath every A and a C underneath every G and so forth? This is just an efficiency thing. So if I ever write in this class just a single string of nucleotides, probably a good assumption that it's a double helix, that I'm just not telling you that there's another strand hydrogen bonded to the strand that I've printed on this screen. So which base pairs, is it AT or CG that has more hydrogen bonds?
GC.
Okay, so GC has, zoom back in, 3 hydrogen bonds. A's and T's have 2. So that's just an example. So then the last thing you need to do to figure this out is what? There's a relationship you have to know to answer this question. What's the relationship you need to know? Something about temperature and base pairs. How does temperature influence how tightly?
Increasing temperature increases denaturing.
So increasing temperature causes the 2 nucleotides to want to separate. So which 2 nucleotide pairs hold together more strongly when they get heated up? Cytosine and guanine, because? Right, so more hydrogen bonds means stronger, which means they're more resistant to the effects of increasing temperature. They hold together tighter. There's more electronic forces holding the G's and C's together because there are more hydrogen bonds. So then, what's the easy way to figure out? These are all exactly the same length. These are all 10 nucleotides long. So which one is going to melt or fall apart first? The one with more A's and T's, which is number 2. Does anybody disagree? I didn't do it, I just wrote the problem. 1, 2, 3, 4, 5, 6, 7 A's and T's. So if you can beat that, if there were 8 or 9 or 10, that would be the weakest double helix in terms of its resistance to being heated up. So any questions about relationships? And you can always come later after you've had a chance to think about, I know this is rapid fire, this is lecture style, we're talking. If you go home or wherever and you start thinking about this and have questions, ask later, absolutely. So here are the 4 really just key take-home points. Hopefully everyone's taking notes. By the way, one of the comments I got on one of the surveys was being unsure about this class because of all the computer work. And one of you, I think, if you're here today, specifically said worried about something about taking notes on the laptop, tablet, smartphone, whatever. I don't demand that you do or don't take notes however you want. Well, I demand nothing. I demand excellence. I demand that you have fun. But take notes however you want. Literally, it doesn't matter. Whatever works best for you, as long as it's not disruptive to the class. I guess I can't give a blanket, interpretive dance your notes or something. No, maybe not. Go outside if you want to go interpretive dance your notes. Okay. But these are the 4 key points then from today. Right? The polarity is important for you to be able to figure out the influence of temperature on whether or not DNA strands are separated or if the hydrogen bonds are formed when it's cooler. and then the base pairing and the number of hydrogen bonds between the nucleotides. This should all likely, but not necessarily, be review. So some of you are yawning, that's fine. Carb coma after lunch, I hope. And that's okay. But one of my jobs, as I'm sure you appreciate from having been in school for most of your life, is that I have to make sure that everybody's up to speed. So for those of you that are advanced, and this is the fourth time you've heard this, I do strive during the semester to try to build in extra activities for you to work on while we're doing these sorts of things. So look for those opportunities as it comes. The other thing I'm going to do for students that have, this is the third or fourth time they might have heard some of this, when I ask you to turn to a partner and talk to things, turn to multiple partners and see if they're all giving you the same answer, because you can help me be the teachers and help all of us get up to speed. This was part of one of the readings that I asked you to look at. An original scientific research paper, Chargaff et al. In 1951. Don't remember dates or names, I don't care. You can look this stuff up if you need to. I don't need you to memorize that for exams. Does anybody have any questions about this? I'm not going to go over it if it's either clear or you didn't take the time to look at it, because we don't need to walk through it if you don't have any questions.
Why would you need a molar ratio for the first two?
For adenine to guanine and thymine to cytosine. So what was the, for those of you that read this, if you didn't read this, what was the point of this paper? What were they doing? What was Chargaff et al.?
I mean, I know that they're comparing like 4 different specimens.
Okay.
And I just kind of knew like the point was just to show that adenine and thymine had like a 1-to-1 molar ratio.
Okay, so this is what wound up being the basis of what we call Chargaff's rule, which is that if you look in any organism, well, actually, I should clarify that. If you look in most organisms, if base pairing is true, if A's always pair with T's and G's always pair with C's, then if you take all of the DNA out of an organism, like one of these 4 that he studied, if you take the mass of all the A's, it should be equal to the mass of all of the T's. Because there should be an equal number of A's and T's. So if you actually extract the DNA and weigh it, it should be the same, or almost the same. And so that's where the point now is being made. Well, sure, we can look at the G to C ratio and see that those ratios and the ratios just above them, regardless of which species Chargaff et al. Looked at, the A to T ratio here in this line was almost always almost 1. 1.02, actually it was 1.02 for all 4 of these. And if you look at G to C, it's 1.02, 0.951, right? This was, they didn't know before, Chargaff, nobody knew before this study that this was true. This was an open-ended experiment. They just said, okay, let's take the DNA out and weigh it and see if we can figure out something about DNA. This was before the structure of DNA was known. They didn't know about base pairing or double-stranded or anything like that. So the question then, coming back to the question was, why did they look at adenine to guanine and thymine to cytosine ratios? Should there be a relationship between those two? Thymine to cytosine, T pairs with A and C pairs with G. So we don't necessarily expect there to be a relationship. And is there? Well, it's definitely not 1. All these numbers down here are 1.8, 1.7, 1.6. So there's a bigger range. The reason that they reported this is because they didn't know. They were testing a hypothesis. So there might be a relationship between, what if DNA base pairs were AG and CT? This experiment would have told them. We would have seen ratios of 1 in those 2 rows, but not those 2 rows. So they were just testing all possible combinations of nucleotides. Well, actually, not all possible combinations, because they did already know that, well, suffice it to say, they did not test all the combinations. These were the 4 critical ones. Good question. All right. So in case there are any questions, I want to spend maybe about 5 minutes-ish on this, if possible, if needed. So for those of you that got into this spreadsheet analysis, this is one of the reasons that it's good to have tablets, smartphones, laptops in class. and outside of class so you can work on these sorts of exercises. So these are actual data on, again, percentages of A, T, G, and C in the genomes of these different species. So we've got E. coli, a bacteria, Diplococcus pneumoniae, which is also a bacteria, causes pneumonia. The bacterium that causes tuberculosis, anyone knows Saccharomyces? Yeast, that's baker's yeast. Paracentrotus is sea urchin, rat, a worm, and whatever sequence you made up as part of this exercise. So one of the questions we had was, C. Briggsy. We'll get there in just a second. I want to make sure that we work on filling in those two yellow spots first, if anybody wants me to go through that. Calculation, using a spreadsheet to calculate the ratio of %A to %T. Does anybody want me to walk through that? Okay, this is pretty quick. So spreadsheet, you can either do one of two things. You can either type in the numbers themselves, like I could type in, so %A goes on the numerator, 29% divided by %T would be 28%. So in Excel or Google Sheets, or probably Numbers, if you're a Mac person and you don't want to use Google or Microsoft products, everything starts with an equals sign, and then I could do 29, which is the %A, slash divided by the 28, which is the %T. And if I hit return, that gives me an answer. It's also 1.03. And one more minute for those of you that are already past this. And if you have more questions about this after class, happy to go through this in more detail if you want. So that's one way to do it, just in any cell, equal sign, whatever mathematical expression you want. What's the better way to do this for those of you that know how to use Excel and Google Sheets better? Instead of typing in the numbers, they're already on the spreadsheet somewhere. So I can type an equal sign, and then I can say I want %A, that's that cell. So if I click on that cell with the mouse, then it enters cell reference B7 right there. So I don't have to retype the numbers. I just say you look in that cell for the numerator, slash, and then what do? Click on C7, hit return, same number. The reason that's better is because if you grab on the lower right-hand corner of a cell that's highlighted that has an equation, and you drag that up the column or down a column, it fills in that equation. So it does that calculation, gives you the answer, that calculation gives you the answer all at once. So if you haven't seen that before, that's why we do these sorts of things in class. It's faster to do lots of calculations. I'm going to move on from there to one other point about this spreadsheet. Again, if you want more help with that, I'm happy to give it. So one of the questions was, C. Briggsy, how do you go from, so that's the research organism I work on. You heard me talk about last time. I gave you one piece of information. They have 30% A. I did that on purpose. It's not a trick question, but from that one piece of information, you can figure the rest of this out. So half a minute, think about it, and then I'm going to ask you how to do this, if you don't already know how. Yeah, please talk to each other if you like. Absolutely. Okay. I heard one of my favorite sounds. It came from the back somewhere. It was, oh. So thank you for that. How do you proceed? You've got 30% of the nucleotides in the genome are A's. And And we know what comes next in the logic. If it's 30% A, it has to be 30% T. For every A, there's a T on the opposite strand. Okay, so that's 60%. How many percent nucleotides are left? 40%, and they have to be split evenly between the remaining two categories so that it totals 100%. And then you could calculate the percent A to percent T ratio, which of course, in this made-up example is 30 over 31 and 20 over 20, which is also 1. Now, one final point before we move into wrapping up. The HTLV, if you clicked that link and you read the paper, they reported percent A, I think, what was percent U? Right, so you can do percent A there, percent U there, percent G there, and percent C there. Did anybody do this? I don't mind if you didn't, but did that match the expectation that were there an equal percentage of A and T, or A and U?
No.
What were the actual numbers, if you know them?
18.3 and 34.9 for G to C.
Oh, sorry.
Oh, yeah, just open there.
So what was the percent A?
23.6.
23.6, and then
23.2.
23.2.
18.3.
18.3.
And 34.9.
34.9. So here's the part where I tell you that a rule has been broken, a rule of biology, and there's a really easy reason why this is. So what is weird about this? The A to T ratio, or A to U ratio, 23.6 divided by 23.2, it's about 1. What about G to C? There are twice as many C's as there are G's. So why does HTLV break this rule? The Chargaff's rule, there should be an equal number of A's and T's and an equal number of G's and C's. It has entirely to do with the V part of HTLV. It's a virus. It has a single-stranded genome. That is, there's no base pairing involved. So the numbers of A's, T's, and G's and C's are totally arbitrary because the sequence of its genome could be, let's see, if there are twice as many C's as G's, it could be C, 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 G, G, right? That's twice as many C's as G's, and then an intermediate number of, say, 3 A's and 3 T's. Right. Uneven number of C's and G's, and by chance, an equal number of A's and T's. So it's just because it's a single-stranded genome, there's no base pairing, there's no second strand, so Chargaff's rule doesn't apply. Yeah?
Would that be considered an RNA, or does it have that
I
OH group?
don't remember does anybody remember if HTLV is an RNA virus or a DNA virus? Probably an RNA virus. Ooh, I know. What clue do we have that it's an RNA virus? The U, which was, mentioned early in class. So if it's got uracil instead of thymine, it's an RNA genome. Great, thank you for asking. Okay, so by now, that's sort of how every class might go. You come with questions, work on the homework assignments. I'll probably design a few things, new exercises to stick in. We'll ask questions, answer questions, do small group work maybe. And now we've covered all 3 of those tasks. So in the last couple of minutes, I want to make a few points, just feedback from last class. Google Classroom and the informed consent forms, by far, everybody did a great job. There were a few of you that I made comments on that somehow a PDF got turned into a Google Sheet or a Google Doc or a Word file and then sent back. If you got those comments, please correct them. See the Discovery Hub for help with that, or ask me if you want. Okay, but otherwise, it went actually way better than normal. So you're all either digital natives or you're geniuses. I'm not sure which. We'll find out. Okay, now I want to give some anonymous feedback and answer a couple of your questions and concerns that came up in the surveys that were due at the beginning of class today. I looked at these last night, so I didn't get the most recent submissions. But there were some trends, so I just want to let you know kind of where we're going in the class and dispel a couple of myths, maybe, that I saw in some of these anonymous comments. So I was actually particularly impressed. A lot of what I heard you wanted to learn about was concepts or topics I talked about last class, which is fine. But there were some broad categories like I want to learn about epigenetics, diseases, gene regulation, how DNA is synthesized or replicated, a lot of heredity and inheritance, how traits are passed from generation to generation. And this quote, which was one of many people that said, I want to learn about mutations and how DNA sequence changes happen, right? And this is very interesting to me too, and I hope to pass this trait on to the rest of you, that it's amazing that a single mutation could have a really huge impact on your life, like getting breast cancer or not, right? That's what it comes down to, essentially. Then there were some specific topics, CRISPR, or gene editing. We will definitely be talking about in class. Genetically modified organisms we'll talk about in class. The genetics of deafness we will actually talk about in class. I had already planned that before I got this comment, but there is one case study we'll look at, a published study on the genetic basis of deafness when we get to the inheritance part of this semester. The probability of obtaining a certain trait, much to many of your chagrins, we will do a little bit of probability and statistics. It's not going to be hard. It's going to be pretty straightforward. I'm going to help you as much as I can get through that because it's really important for genetics. Myths and misconceptions with genetics. From time to time, we'll get into some interesting stories about beliefs that people have about genetics that aren't true, and vice versa. Neurodegeneration, we'll talk about the genetics of some neurodegenerative disorders. And most surprisingly to me, somebody wants to learn more about the Hox genes, which I find fascinating also, but I'm not sure we'll specifically cover those. That's, if you want more information on Hox genes, if we don't get it this semester, take developmental biology, 155. Okay, last couple of points. You were probably not surprised to see these two comments. When I asked, what do you expect to learn about you don't want to learn about? I got a lot of, I don't want to know about this because, this is like the fourth time I would have heard this, mitosis, meiosis, what we just talked about, ATGC, hydrogen bonds, polarity. Or there was a lot of, I've struggled with this topic in the past, which is absolutely understandable. And then here are more specifics. So mapping genes we won't do so much of in class. We will talk about DNA sequencing because it's a modern technique that underlies most modern genetic analysis. We will be doing some chi-square tests. I promise you, if you don't think you like or understand chi-square tests, I come at teaching statistics and probability and genetics in a way you've probably never seen, and I hope it works for you, because sometimes it's just that you've heard it delivered the same way over and over and over again. And of course that doesn't work. You need something fresh. So I'm going to try to do that. Mitosis and meiosis are critical to understanding inheritance. So we will talk about those. But again, I hope I bring a fresh perspective to that. Genotypes, phenotypes, alleles. We won't do so much on crossing over. If you were the person that said you didn't want to learn about that. Protein coding, absolutely. We're going to do central dogma. DNA encodes RNA, which is translated to protein. That's critical. And so this last point I put on here for a very specific reason. So in the last couple minutes, memorizing things is boring to me too. And as I said earlier in class, the reason that we all have these things is because we can look stuff up if we don't want to memorize it. And I want you to take advantage of that. So there will not be a lot of memorization. In fact, that's why I said last class, the exams will be PDFs I send out to you. You've got internet-connected devices. So they're open internet tests. I design tests for you to take advantage of the knowledge that exists on the internet. The only thing I'm going to ask, I'll tell you now and I'll tell you again before the exams, is not to collaborate with each other during tests. So looking things up is okay, but not communicating with each other during exams. Right. So I'll remind you about that as we go. So yeah, we talked about reasons why you didn't want to learn that. Now, my favorite comment, this is great. I love hearing these sorts of things. It only showed up once, but that's fine. So now, lastly, fears. And then I'm going to close with what to do for next class. As always, these slides are on Canvas already. So you've got all these notes already. Time and workload, I will do my best to make sure it's not onerous. And we already talked about, I'm going to try to make the computer part reasonable. I appreciate feedback. So if at any point you feel like I'm being unreasonable about requests or the use of technology, let me know. Okay, I'll cover the rest of these points. You can look over them yourself if you want to. Next class, Have a happy Friday, by the way. But the end of the first manual, the end of the first chapter to read for today, if you didn't do this, please try it for next class. At the end of every chapter, there's a first attempt in learning, failed. The point of these are you try it in advance before we talk about the content to see how far you can get. They're not meant necessarily for you to succeed, so don't spend a ton of time reading it. Try it, see where you get to. When you get stuck, come to class and ask questions about that. Okay? The next course manual chapter, chromosome structure, and then 2 more short surveys, please. The last 2 surveys for the start of the semester, and those are available as assignments on Canvas already as well. Thanks very much. I'll hang out a bit if there are questions, and have great weekends.